Okay. Good morning, students. Today we are going to learn about the summary of Unit 1, starting from the definition of biology to the laboratory safety rules. When we come to the first session, biology has a relationship with chemistry, physics through the students of the study of elements, chemical bonds, and light energy. Other scientific concepts like conversion of radiant energy into organic molecules by photosynthesis is studied with the knowledge of chemistry and physics. This we have learned in the previous one. Living things are different from non-living things because living things have some common properties. All living things have, in commonly, they have properties. Those properties are shared by all living things or living organisms. Everything can be considered as to be alive if it displays all of the important properties simultaneously. Living systems have common characteristics. They are composed of living things are composed of one or more cells. Living things are one cells. For example, yeast cell under the fungi, and complex more cells are higher organisms. For example, like me, I am 100 billion collections of cells. So, living things are quite complex and highly ordered, can respond to stimuli. If there is touch, if there is light, if there is anything, living things can respond to the environmental stimuli. The other main characteristics which is shared by all living organisms is they can grow. Living things can grow. They can also reproduce each other and transmit. When they reproduce, they transmit the genetic information to their newly existing offsprings. Need energy. To do any activity, living things need what? Energy to accomplish their tasks, and they can relatively contain constant internal conditions, that is homeostasis, and they are what? Capable of evolutionary adaptation to the newly environment. If there is harsh environment or suitable environment, living things can accommodate the condition in order to cope up with the newly existing environment. So, living systems show hierarchical organizations. The hierarchical organization of living systems progress from atom to the biosphere. Living things, hierarchical organization starts from atoms to a biosphere. At each higher level, emergent properties arise that are greater than the sum of the parts. Thus, biology unifies much of natural science. The study of biological system is interdisciplinary because solutions require many different approaches to solve a problem. For example, biology also studies the nature of energy transactions by thermodynamics. You have learned in high school chemistry, that is what? Thermodynamics, and the conversion of radiant energy into organic molecules by photosynthesis. The reactions known in chemistry and physics. Living things are what? As we have learned in the previous one, they are composed of what? One or more cells. They are complex and highly ordered. They can respond to stimuli. They can grow. They can reproduce. They can transmit their genetic information to their offspring. To do cellular activities, they need energy to accomplish their activities. They can maintain relatively constant internal conditions. That is what? Homeostasis, in other words. They are capable evolutionary adaptation to the environment. newly environment. Okay, now let us see the application of studying biology. What is the advantage of studying biology? The advantage of studying biology here is, for example, to make wine, or the brewing of beer, baking of bread, and the production of cheese all depend on fermentation process brought about by yeasts, other fungi and bacteria or enzymes from these microorganisms or single-celled organisms. The other application of biology is production of antibiotics such as what? Penicillin, which are produced by mold, fungi, or bacteria. The production of industrial chemicals such as citric acid or lactic acid needs bacteria or fungi to bring about essential chemical changes. When our environment is disposed or polluted by contaminants, we use application of biology, that is sewage disposal depend on bacteria, and the filter depend to form the base of the food chain that purifies the effluent. The other application of biology is radiation. Radiation can also be medically beneficial, which is helping to treat cancer. We use radiation. The radiation can treat the cancer patient studied by combination of two subjects, that is biology and physics. In biology, there is the cornerstone of biology is the structure of DNA. Those described or modified by two scientists also are Watson and Crick in 1953, and subsequent achievements in DNA science lead to the technology of DNA manipulation that are transforming applied fields such as medicine. DNA can be applied in medicine, in agriculture. DNA in agriculture, how DNA is used in medicine, how DNA is used in agriculture, and how DNA is applied in forensic science. We will discuss this in detail in biotechnology sessions. And the other is forensic analysis of DNA samples from crime scenes. The next section of this unit 1 is the main tools of biologists, that is microscope. The microscope have several different powerful lenses attached to them, allowing the viewer or the scientist or the biologist to what? To inspect the content at more than 100 times its actual size. Biology, as you know, it is a modern science which could not exist without the microscope. And good microscopes are essential for a day-to-day activities for most biology. In addition to their importance, microscopes are extremely expensive. We cannot find microscope in a simple way, so we have to give care for our microscopes. Therefore, it is important to handle the device properly. How to handle the microscope? The microscope in this section, microscope had handling such as removing it from a cabinet to do any activities, carrying and preparing it. Carrying, when you transfer the microscope from the cabinet into the bench, there is a way how to handle this microscope. And preparing, how to prepare the microscope for use or for observing cellular organisms. Focusing will be elaborated and should you practiced by all sessions. How to control and focus the light. In microscope activity, controlling and focusing is the main activities. Observing from the side of the microscope, use the coarse focus. Coarse focus, as we have learned, the parts of the microscope. To open focus on your microscope until the stage and objective lens are as far as possible. The turn the illuminator and adjust to maximum brightness. We have to adjust the brightness of our microscope. The other one is place a piece of thin paper on the stage. On the stage, we have to place the thin paper placed to cover the hole in the stage. That is what? In the previous, we have said that aperture. Aperture is the stage hole part of the microscope. Again, as a stage, using the stage clip or mechanical stage. We use a mechanical stage. A bright illuminated circle should be what? Visible through the paper. If our substage condenser is focusable, rack the focus up and down and note the effect on the size and sharpness of the illuminated circle on the paper. Look at the diaphragm control. The diaphragm, as we have learned in the previous, diaphragm is it is a part of microscope that controls the entering or the amount of light. Adjust the control to open the diaphragm. If you microscope has a disc diaphragm, rotate the disc. Use the diameter so to decrease the brightness and then back to the microscope. Now let us come to the focusing part of the microscope while using it. We observe the slide starting from the low power. First, every activity in the observation of a specimen through a microscope starts from what? The low power objective, that is 4x in position. The other objective is 10x objective is longer, the 40x objective longer, and the highest 100x or the sample is magnified 100 times, objective longest of all. Gently turn the coarse focus knob to close focus until you reach the focus stop. Now look through the eyepiece. Look through the eyepiece. Use the coarse focus knob until you get the sharp focus. Use the focus knob to squeeze the focus. Note that as one part of the field of view came into the sharp, depth of the focus decrease with increasing magnification. A specimen that looks planar at 40 times A or 40x may show depth at higher magnification. You know, magnification is the ratio of the size of the image to the size of the object. Magnification is the ratio of the size of the image to the size of what? Our sample or our object. We can find the magnification, that is size of our image over size of our object or our sample. With a compound microscope, the magnification is what? The product of both the eyepiece and objective lens. So if a microscope has a 10x eyepiece and 40x objective lens, the total magnification is 10 times 40, that is 400. That is the size of our enlarged sample. Images of the structure seen with the microscope usually show give the magnification so that the actual size of an object can be calculated based on the formula. Actual size of glomerulus is equal to size of image over magnification. Okay, now let me ask you a question. What is the difference between light microscope and electron microscope? Light microscope uses a beam of light to view specimens, and in the case of electron microscope uses a beam of electrons instead of a beam of light, and electromagnets instead of glass lenses to enlarge the image of our samples. The other question, the second question is, what is the function of test tubes, wash bottle, dropper, or list the biological laboratory equipment and their functions. Let me tell you some of them. Test tube. The use of test tube is to culture microorganisms. We put our samples and give the culture components, and we find the desired amount of our microorganisms. The other is wash bottle. What is the use of wash bottle? To wash and scrub glassware. Dropper. Dropper is to transfer a small volume liquids. That is drop by drop. Test tube rack to hold our test tubes. Spatula, as we have learned in the previous, to pick a laboratory chemicals or media from a stuck container. Dissecting pan, we use dissecting pan in order not to contaminate our desks from the dissecting organisms. Mortar and pestle to grind substances. Forceps to hold up or pick up small materials. Okay, the other forceps to hold up or pick up small materials. When we do not or we cannot hold or pick up small materials with our fingers, we use forceps instead. Another one is flasks, which is used for culturing, heating substances, for temporary storage of solutions and liquid specimens. The other is microscope, which is the main tools of biologists. Objective lens to facilitate a quick change in the magnification of a specimen, and a high or objective lens provide a higher degree of magnification, which allows to zoom in close to the object being studied and see more detail, and a lower power lens will provide a wider field of vision. The diaphragm on the microscope is used to change the amount of light. That is being allowed to enter through the slide. Closing the diaphragm will decrease the amount of illumination of the specimen, but increases the amount of contrast. The light to the field is reduced as the diaphragm is closed. Similar diameter, and therefore let us in less light. In order to focus on an object, one should move the stage away from the objective lens rather than towards each other in order to prevent crashing the lens into the slide. We use, for higher magnification, oil immersion technique used to increase the resolving power of a microscope and improve the image quality. The oil immersion increase the resolving power by decrease the light refraction, bending of the light, allowing more light to pass through the specimen to the objective lens. Oil immersion eliminate any air gaps and loss of light due to refraction and maximizes the amount of light that goes through the objective lens. Now it is enough for today. We will meet on the Unit 2 session in the next video. If you find this video important, subscribe, share, and like. Thank you for watching.